Today we have a presentation by Kūpono. Kūpono is a graduate student at the University of Hawaii at Hilo. His presentation is titled Seed Dispersal by Wild Pigs in Hawaiian Forests. His research is connected through a permit with the HTF. Please note the permits are required when entering HTF for research purposes. Kūpono, you may start whenever you're ready.
All right, mahalo nui for that introduction, and mahalo everyone for coming and sharing space with me today to learn about wild pigs and seed dispersal. So just a little bit about myself. My name is Kūpono Aguirre. I'm from Waikapu, Maui. I was born and raised on Maui, and I moved to Hilo in 2018 to attend UH Hilo. My undergrad is in environmental studies, and I'm currently a master's student in the Tropical Conservation Biology and Environmental Science program at UH Hilo as well. So to start off, just a little bit of background. Growing up, I've been around pigs ever since I can remember. My great-grandparents had a pig farm in Ha'iku on Maui, and they raised hundreds of pigs. I even had a pet pig growing up. She's going to be 7 this year. And one of the cool things that I noticed about having her is that when we would feed her pumpkins, tomatoes, zucchini, stuff like that, my grandfather uses her manure for the garden. So we'd compost it, and a lot of those things started sprouting up in the compost. So I thought that was really cool. That kind of has been in the back of my mind since high school. And in undergrad, I actually did some research on non-native seed dispersal of birds that were, or non-native birds, sorry, here in Hawaii. And I thought, why not pigs? You know, pigs are spreading seeds, and maybe I should check out what they're spreading. So yeah, that's kind of the inspiration behind my project. I'll go into it a little bit more throughout my presentation, but yeah, that's just some background on myself. Okay, so just a general overview. First of all, I'll talk about the arrival of pigs here in Hawaii, then my inspiration for the project, as well as how I explored my methods, the outcomes of those methods and exploration, as well as some takeaways. And then I believe at the end, we will have time for questions. So firstly, the arrival of pigs. So they were first introduced here in Hawaii by early Polynesian settlers, and they were basically brought here as a food source. They were prized for ceremonies and special occasions, and were not consumed necessarily every day, but more so for important events or to help boost strength for war. These early pigs were actually a lot smaller in size, and they were kept near homes or in like rudimentary type pens near the houses. They were not in the forests due to the lack of food resources in the forest at the time. Pigs really like things like sweet, fleshy fruit and earthworms, and those were not introduced yet, so the pigs were not able to colonize the forests. After the later introductions of these non-native fruit species and earthworms, the pigs were then able to move into forest settings, as well as hybridize with other domestic breeds that were much larger, as well as Asiatic and European wild boar. So currently, as a result of this intermixing, the pigs are well established in forests throughout Hawaii. They're much larger, like I said, than the smaller counterparts that were first introduced by Polynesians. So the current pigs today have about 25% Polynesian ancestry that were original descendants from the first pigs brought by Polynesians. So our current understanding of wild pigs in Hawaii is that they have detrimental negative impacts on forest ecosystems. Through their trampling and rooting in the soils, they erode and compact them, as well as damaging and eating native plants in the process. They also prey on ground nesting birds such as nene and their eggs, as well as seabirds that nest in coastal areas. And then their rooting and wallowing also creates small puddles and wells in hapu'u and in the ground that can be grounds for breeding mosquitoes, which spread avian malaria. So the inspiration behind my thesis. So this is the oma'o, which is a native thrush found here on Hawaii Island, and it's actually the last functional native frugivorous bird. We do have the puaohi on Kauai, but unfortunately its numbers are too low to where it's not considered a functional seed disperser. We do have non-native bird species, but they do not fulfill the role of native seed dispersal fully in the way that oma'o and other native frugivores do. I also looked at some other studies across the Pacific. One done in Aotearoa specifically found that a single tree species, matai, which is culturally and ecologically important to Aotearoa, was dispersed by pigs in both captive feeding trials and in wild collected scat. So from scat that was germinated from these samples, they found that actually, I think it was actually hundreds, if not thousands, of these seeds that were found in the scat samples, showing that the pigs were eating a lot of them. However, this was the only species that was found in these samples. There were no other intact seeds recovered. So it just shows that they were feasting on one type of species in this area. It just shows that they kind of had preference here in Aotearoa. And then in Guam, because of the brown tree snakes, which have just decimated the bird populations, this study was done to focus on pigs and deer and see what kinds of seeds they were dispersing and how they were damaging the forests there. They found that the native tree Morinda citrifolia, also known as noni here in Hawaii, was actually dispersed by pigs, as well as a few other native species, as well as non-native species. All right, so the exploration of my project, going into my methods for the field and the greenhouse. So first of all, I want to talk a little bit about my approach to my methods. So I wanted to look at what pigs are dispersing. So we currently know that pigs are eating non-native fruits. I wanted to see, are they also dispersing native seeds as well, such as those studies that we saw in Aotearoa and in Guam. So in order to do this, I needed to categorize the vegetation first to see what was out there in the forest. And then once I determined what kinds of species were there, I looked at the fruiting species specifically. And once I went to these fruiting species, I needed to find a way to quantify the availability of fruit in the forest based off of the species that I had currently surveyed. The best part of this is scatology, which I just learned the word for this a couple of days ago. It's basically the study of scat or feces and how that affects different processes. And for me specifically, I'm looking at seed dispersal in scat. So I have to identify and be able to tell pig scat from other species. So this was a little portion of my project as well. Also in determining preference. So I wanted to see, do they prefer certain fruits? Just like in Aotearoa, where they found that they were consuming mostly matai in that area. I wanted to see, are they consuming certain species based off of what's there in the forest? Or are they selecting for certain fruits based off of their own preferences? To do this, I only looked at woody fruiting species and some herbaceous species that had fruit as well. However, I did not look at any grasses or like weeds and common greenhouse weeds. Okay, so for selecting my sites, I wanted to look across an elevational gradient and see whether there were differences between high elevation and low elevation sites. So I'll go into a little bit more about my study sites and my selections for elevation later on. I was gonna have 4 collections total for each season, but because it's not very easy to find pig scat sometimes, it got pushed back a little. I do have 4 collections total, but they're not completely in sync with each of the seasons. So there is some variation in collection, but I do have 2 collections done so far, including the grow out period. So you're kind of seeing I'm halfway there. So these are my study sites that I selected. I selected 3 low elevation sites, which are Keaukaha Military Reservation, Keauhana Forest Reserve, and Waiakea Forest Reserve, which is up Steinbach. So these are my 3 low elevation sites, as you can see on the right of this map. And then to the left of this map, we have Gila Watershed Reserve, which is up Saddle, Lapahoehoe, which is through the HETF permit that I was allowed to access, and the Hakalau Forest National Wildlife Refuge. So as you can see, most of these are within the same areas within Hilo and the Puna area, but I tried to keep it within the same amount of rainfall, so they're all generally considered wet forests. So for my vegetation surveys, for both vegetation and phenology, I used a 1 kilometer transect, but to do the vegetation surveys themselves, I used a combination of point intercept and brown blanquet with a 5 meter radius for each circular plot. And then I basically continued doing these plots until no new species were found in 2 consecutive plots. So once the vegetation was determined, I then went and took 5 random points along my transect. So each collection, I took 5 new random points for a total of 20 random points. And at each point, I'd find the nearest adult tree, and then I'd classify the fruit cover based on the percentage of branch tips with available mature fruit on them. For my greenhouse methods, I used 10 by 20-inch seedling trays with the wooden cedar inserts, as you can see on the right, to create 6 different compartments for each tray. My media mixture consisted of a 1-to-1 ratio of perlite and peat moss. The samples were misted once daily for 5 minutes in the morning time. And that was adjusted a little bit during the summer when it was drier and I noticed that the samples were drying out. So I would increase or decrease the minutes based on the available moisture. Once the germinants were large enough to be able to be identified, I then removed them from the media and counted them. So I had like a little hand clicker and I was basically counting how many seedlings of each species there were that were able to germinate from the scat. After each collection, the total grow out period was 6 months. So my last collection was last month. So I will be done by July with the grow outs. All right, so the outcomes of my exploration. As you can see on the left-hand side here, this is just something cool I wanted to point out. The top picture is a picture of a pig scat that I found in the forest. And then the bottom picture is in that same area on that same trail. It's another pig scat that is covered in what those are actually Cecropia seedlings. So as you can see, I don't know if you can see my mouse, but in the upper left-hand photo, those smaller seeds are actually all Cecropia seeds. These bigger ones are strawberry guava seeds, and then this one here, I don't know if you can see that, but some of them are actually lilikoi seeds or passion fruit seeds. So you can actually see the seeds in the scat when you're going out to collect them, and then you'll even see some samples like this that are already germinated. But for the sake of my project, I was only collecting the ones like you could see above. I wasn't collecting anything that was already germinated, and anything less than the size of my palm, I didn't collect either. So this is what happened. At first I was a little worried that nothing would sprout from the scat, but then I quickly realized that my problem was that too much was sprouting from the scat. On the left-hand side here, you can see tons of seedlings in all of the trays, and this happens within a matter of weeks. On the right side, these are all Cecropia seedlings that have completely overtaken the germination trays. And unfortunately, once it reaches the point where the roots have completely compacted within the tray, there's not much else I can do to try to germinate the rest of the seeds. What I did do was I cut off the tops of the seedlings, and then I tried to remix the roots and the media back in with more media to try to germinate any seeds that were still in there. Unfortunately, that didn't necessarily work, and nothing really germinated after that, which just shows how invasive some of these plant species are when they do germinate from the scat. So yeah, that's a Cecropia on the right side. These are all the species so far that germinated from the scat. I'll just take a little moment for you folks to look through this here. Some of the really cool ones that I didn't expect were strawberries. That was really cool to me. And then the closest thing to a native, it's not native, but it's Polynesian introduced, is the noni, the Morinda citrifolia. I thought that was really cool because in the Guam study, that's one of the species that they found that pigs were spreading over there. So it's cool to see that here in Hawaii, pigs are also spreading that species. And then these are just some photos of some of the seedlings that I wanted to include just to see. They're kind of cool. On the left side, we have Java plums. The right side is Clydemia with the little fuzzy hairs in the middle. And then we have blackberry on the left, and then the strawberries on the right. These are just some of the seedlings. But yeah, so this blackberry on the left is actually from Lapu hoi hoi. I got hundreds of them, which I'm going to cover in a little bit. So from my first collection, which occurred in January and December, you can see that, okay, so the gray is strawberry guava. It's over 75% strawberry guava. The orange is the yellow is common guava. The blue is cecropia, and then the orange is passion fruit. So you can see that the majority of the samples are actually, of this sample is strawberry guava and common guava. And then in my second collection, April and May, there was no common guava at all. The strawberry guava dropped from about 75% to 25%. And then the Cecropia just blew up to over half of the samples. And here we see we have about 25% blackberry, which was not seen in the first collection. There were other species that sprouted in these collections, but there were so few of them that they were, when calculating the total percentages for the germinants, they were less than 0%, so they weren't showing up on the graphs anyway, but I did include them in the species list. So I had a series of hypotheses that I wanted to test for my project. Many of them dealt with species richness and the relative abundance of germinants in the scat. So the species richness is basically just the total number of species that I found in any given scat sample. I categorize these by both site and by elevation. So I have the number of species as well as the number of individuals for each species by sites and by high versus low. So my first hypothesis deals with the species richness in the forest, which is the amount of fruiting species that I counted that were out there as adult trees, and then the amount of species that I counted that germinated from the scat. So to test this, I did a paired t-test, and the results of that test were there was no difference between the species richness in the forest and in the scat. So you can see they're pretty similar. So Hakalau actually has 0 in the forest because I only counted fruiting species and or species that had fleshy fruits that the pigs would be eating, and it was mostly just koa and gorse, which I wasn't counting because they didn't have any fruit. But as you can see in these other samples, a lot of them, it seems like they have higher species richness in the forest, but you have to think about the fact that they're eating different things that aren't necessarily there. So that while the species in the forest might be greater, the species in the scat could be less, but they could be a little bit different. So it's not really considering the fact that they're the same species, but they're like a lot different. So this is for collection number 2. I combined the two collections for my analysis, so I still didn't find any significant difference between the richness in the forest and the scat. My second hypothesis was also with species richness, but it dealt with only the scat and it dealt with high and low elevations. So my expectation for this is because lower elevation sites tend to be dominated by invasive species because they're highly invaded, whereas upper elevation high sites tend to be more native dominant. And I expected to find more native species at the higher sites and more non-native species at the lower sites. However, I did not find any native species at all, so it kind of skewed my results and I have to reassess. So the outcome of this was that there was no difference between the high and low elevation sites. Despite the fact that there were different species at both of these sites, there were kind of core species that were found in the scat that weren't necessarily what was abundant in the forest, but it was abundant in the scat, suggesting that pigs are selecting for certain species that they prefer over others, regardless of elevation or site. So this last one deals with percent cover of fruit as well as the relative abundance of germinants in the scat, and then comparing that to high versus low elevations. So the reasoning behind this is that we already observed preference for sweet, fleshy fruit that are non-native. And I just wanted to see if there was a difference between what was available in the forest versus what they were eating. So for this one, I found no significant relationship between elevation and relative abundance of germinates in the scat and location, meaning was it found in the forest or was it found in the scat? So that was it basically told me right then and there that they're choosing what they prefer. And what they prefer are these sweet fleshy fruits like guava, cecropia, common guava, blackberry. These are the things that they want. And so they're going to seek these things out regardless of what's in the forest, because it just shows that there's high amounts of these species that are germinating from the seedlings, even when they're not necessarily found in the areas that I surveyed. So some of the takeaways from my project that I want you folks to know. So feral pigs so far have not been shown to disperse native seeds here in Hawaii. The fruit availability also changes throughout the year, as we saw with the blackberries only not even showing up on the graph to being 25% of the graph, and then cecropia from being not even 5% of the graph to over half of the graph. It's crazy to see that at certain times of the year when certain fruits are available, they're just gorging themselves on those fruits. And then when other fruits become available, they kind of move on from food source to food source. So there was no significant relationship between species richness in the scat and elevation or fruit cover in the forest. So they're not eating what's there, and it doesn't matter what elevation. They're eating what they want to eat. Yeah, so we found that preference, and they also differ from non-native birds in that they don't spread native seeds like non-native birds do. When it comes to conservation, some of the things that we want to consider for this are that pigs can spread invasive species like guava, like lilikoi, into areas that they previously were not found in. And one of the big key things of that is that they could be excluded from areas that are more sensitive that don't already have these invasive species, because according to what I've found so far, they can actually bring these species in from other areas that they're getting these invasives from, and then bringing them to areas that they're currently not found. Oh yeah. And then one of the other key things that I found in my research of other papers is that I found a paper from the 80s done on Maui that found that when pigs are not eating strawberry guava, they turn to other food sources. So removing these non-native fruits could put extra pressure on other food sources, or be pushing pigs to eat things like hopu'u, more earthworms, grasses, or other non-fruit items, even meat. Okay, yeah. So that is my presentation. I just want to thank all of you for coming and listening today. And if you have any questions, I'd love to answer them.
Mahalo nui Kūpono. That was a really awesome presentation. And, I don't see any questions in the chat. However, anybody who's on right now that wants to chime in, by all means, please either raise your hand or you can drop it in the chat, whatever you feel comfortable doing. Thank you so much, though, Kūpono. It was really great and kind of terrifying also.
Hi, Kūpono. This is Steve Hess.
Hey, Steve.
Nice presentation there.
Thank
Enjoyed
you.
it. I have one rhetorical question for you. Why hasn't anybody done this before in Hawaii? I know Chiang Giang did it on Maui there, but not to the extent that you did it. There's a lot of room for creative new research like what you're doing. It's great. And some of the differences that we see, like between Aotearoa and Guam and Hawaii, might be, Hawaii, we're out here, and the large fleshy fruit, native fleshy fruit, couldn't make it this far. And like even Guam has the native Artocarpus with ulu there, with seeds in it, and the ulu we have doesn't have seeds, for example. But Also, just how your results are looking towards this most recent paper that I saw about, stand level replacement of trees in Hawaii. And, are those some of the same species that, you know, in the pig scat that are, replacing our native forest trees throughout the state? If you want to discuss that.
Yeah, that's actually really interesting. So, one of my study sites at the Keaukaha Military Reserve, we actually have seen Java plums and noni sprouting up in open field areas. And it's those same areas that I'm collecting the pig scat from. And it's super interesting because I have photos I can share with you. I have photos of Java plums and noni sprouting in scat right next to little seedlings of noni and Java plums that are growing in the same field. So I'm I'm sure the stuff that they're spreading can be replacing some of the forest. I don't doubt it just because we see the same thing with strawberry guava, banana polka, all kinds of stuff that I see growing. So that could very well be a factor going into play with the replacement of our forest here in Hawaii. Yeah, I could see that.
Thanks. Good work.
Thank you.
Kūpono, I think Elliot has a question for you or maybe a comment.
Okay, okay.
Great presentation. Excited that you're doing this work. And and apologies if I missed it, but is it that there aren't large, sweet-fruited native species available in the forest, and so the pigs are just going for the best fruits, whether it's native or non-native, or are they preferring non-native fruits? And I guess one thing I'm thinking of is, if for other places on the island, like the Puawa Forest Bird Sanctuary, which has a lot of akala, would there be in places with more native fruits, would you expect that the pigs would be dispersing those as well, or would they still be just preferring the non-natives?
Yeah, thank you for that question. That's a great question. So my expectations were that at these higher elevation, more native dominant sites, there would be native seed dispersal. And I haven't seen that, but that'd be really cool to look at other places where there are greater amounts of native fruit. But yeah, to go back to your point about there not being these big kind of sweet fruits that we have that are native, I think that also plays a big role in it because if I were a pig, I wouldn't want to be eating our native seeds. They're not very delicious. But yeah, so I think a combination of us not having the seeds, pigs preferring these sweet fleshy fruits, and then the lack of natives are all coming into play for what kinds of things pigs are spreading. But yeah, it's super interesting. One of my sites is up at Hakalau, and I actually collected during the season when akala was fruiting, and I was like, yes, there's gonna be akala, there has to be, it's everywhere. And the only thing that I got was a single strawberry plant. So I don't know if they're just not eating enough akala or the birds are getting it first. I don't know. But yeah, that's a really good question. I'd love to explore that more.
To build on that too, just like thinking that some of the places with the most dense amount of natives are also the places where we are already having fences or want to keep maintaining them. So that being like another reason possibly why you're not seeing that heavily of a presence.
Yeah, definitely. Yeah. And I mean, from what I've seen so far with how many invasives they spread, we maybe want to keep it that way for places that are more sensitive that don't already have these invasives there. But yeah, if there are pigs already there, it'd be interesting to see what they are eating.
Yeah, one of my questions for you, Kūpono, is like when you were out at these different sites, were there places that it was a little bit easier for you to find scat samples, or was there more of an abundance at some sites than others?
Definitely. Yes. Sorry I did not touch upon that. But yeah, so my sample sizes varied so much just because some areas I found just excessive amounts of scat everywhere. And then some of my sites I'd have to go back to over and over and over until I found just enough to have something. So yeah, just that also plays a role too, like how many I was able to find. But for the most part, I got a good amount of samples from all of my sites. Have like hundreds of poop collections. But yeah, yeah. Sorry, I didn't really answer your question, but yes, I some sites I found a lot more than others. So it's like it's up to chance. I don't know. That's what I tell people. Sometimes you go out and you find stuff and sometimes you don't. And that's just how it goes.
Micah, you totally did answer that, and no, sorry. Because I was just thinking, like, based on my own experiences at Hawaii, I mentioned this to you earlier, but just like for everyone else, it's kind of really interesting how much, like, banana polka, for example, I was finding and not really like seeing the plants, but definitely seeing the scat and that be
Yeah.
popping up in the scat. So kind of unfortunate that that's what's coming up in the understory, while simultaneously we're having this dieback from like the rod with the ohias. Mahalo.
Well,
And I think oh, Elliot, yeah, you have another one? Go ahead.
I'm wondering, do you have a sense, and maybe Steve's done some work on this, but of the home range size of these pigs and like for an individual, how far, you would expect that seed dispersal radius to be?
okay. Yeah. I've done a little bit of research into that, but I think Steve could probably answer that better than I ever could. Steve, if you could just share a little bit about the home range size.
Yeah, I guess I'd put it into perspective of other animals. And so pigs don't have very big home range sizes. If you think of an animal like a nene, they are traveling top to bottom on the Big Island. They're covering large areas. A feral cat is covering very large areas. Pig is not. And so they're pretty limited. There's some movement studies that show they follow some seasonal resources that maybe Kūpono could tie into with that. But I would say, we know we can fence them into small areas, and they do just fine. They survive. They don't need the resources from outside those limited areas. So I'd say pretty small in the scope of things.
Thank you, Steve. Yeah, I would agree. Like the seasonal variability, I think they definitely could be moving with as the resources change throughout the year. But yeah, it seems like they got to move somewhat because where I was doing vegetation surveys on the same transect, I would like Tristan was sharing, we would find things that weren't in the trees but were in the scat. So it's coming from somewhere.
Yeah, and then just to like build on that, like whether or not there's agricultural lands in that surrounding area, whether it's mostly like a residential area that they're butting up against, I'm sure really changes that as well.
Oh yeah, I would love to go into residential areas because I have a bunch of people that are like, come to my yard, come collect from my yard, come see what's growing there. So that'd be really cool to compare. So yeah, thank you for that.
Awesome. Really informational. So funny that we can be excited about scat and that you have a really holistic view of, that we can interact with these beings and, it's not all or nothing necessarily. We talked about that a little bit before this session, but we do have one question in the chat. Chuck says, is there any chance that pigs were dispersing native strawberry? Was it present in any of the study sites?
Well, if you can identify native strawberries, please help me out because I do have photos of the strawberries. I just assumed they weren't native because I assumed the native one was a lot more rare, but I do have strawberries that popped up. One popped up at Hakalau, and then I do have like a dozen strawberries that popped up from my site along Saddle Road. So if anyone wants to shoot their shot at identifying strawberries, be my guest. I have lots of pictures for you folks, but yeah, I'm not too sure if they're native or not. I just assumed that they weren't the native ones so far from what I've tried to identify myself anyway.
Yeah, Avalon just chimed in. She said she'd love to see him. I guess I'm curious, if you're okay, Kūpono, we can forward your email to folks, if who are interested in following up and having maybe additional questions or, keeping contact with you in the future. So if you're all right with that and anybody
Yeah,
is
definitely.
curious, we can definitely make the connection.
Okay.
And I
Yeah.
do see that you listed your email right there too. So
Yeah.
mahalo.
Perfect. Yeah, I'd love if anyone has questions, just any insight, want to help identify plants, let me know. I'm open to anything.
Awesome. Well, I think that's it. And, we are going to be having another webinar in March, and that will be with Anna Flores, and she's focusing on Puakala, and her talk will be the drought response as plants grow up, simultaneous effects of ontogeny and phenotypic plasticity. So mahalo again, Kūpono, for joining us, and you folks can be all on the lookout for that. And mahalo everyone who attended.
All right. Mahalo, everyone. Mahalo, Tristan. Thank you so much.
Ahui ho. Take care.
We hope.